I want you to imagine just a massive piece of heavy machinery left out in a damp, salty junkyard for like a decade.
Oh yeah, that's not going to end well.
Right. You know exactly what happens. It slowly gets consumed by that flaky orange rust until the metal literally just crumbles into dust.
Exactly.
We all understand that process on a macro scale. But what if I told you that an almost identical chemical reaction is happening inside your own cells, like right now? It's a process that is literally rusting you from the inside out.
Yeah, it's a very vivid image, but chemically speaking, it's not even a metaphor. I mean, it is an incredibly accurate description of a highly destructive, iron-dependent form of cell death.
Well, welcome to the deep dive. I know you're listening because you, like us, love getting into the actual mechanics of how things work. And today, we are looking at something that flips a lot of our textbook biology completely on its head.
It really does. It's a massive paradigm shift.
Yeah. So our grounding source today is a really dense, cutting-edge research paper. It was published in the journal Nature, June 3, 2026. And the title is Spermine is an endogenous iron chelator that inhibits ferroptosis.
Which I know might sound like a really heavy dose of biochemistry right off the bat, but the implications here are just staggering. We are fundamentally redefining how a very common molecule in your body dictates whether a cell lives or dies.
Okay, let's unpack this. The mission today is to understand how this everyday molecule, spermine, acts as this secret molecular sponge for iron.
Right.
We're going to explore how aggressive cancer cells hijack this sponge to make themselves essentially immortal. And conversely, how emergency room doctors might soon use that exact same sponge to save your organs after a massive trauma.
And to really grasp the magnitude of what these researchers discovered, we have to start with that rusting process you mentioned.
Okay, lay it on us.
The scientific term for this is ferroptosis. So this is a unique, non-apoptotic form of cell death. It's entirely driven by lipid peroxidation.
Right, and we all know the classic everyday examples of lipid peroxidation, right? It's the whole sliced apple turning brown on the counter idea, or like when cooking oil goes rancid.
Exactly, yeah.
The fats, or the lipids in the cell membrane, are getting damaged by reactive oxygen species. But what this Nature paper does is zoom in on the specific catalyst for that cellular damage. It's not just ambient oxygen causing the problem. It's rogue, unbound iron just running rampant through the lipid bilayer.
That's the key. That unbound iron is called labile ferrous iron, or F2 plus Xea. And when you have too much of this free iron floating around, it interacts with cellular peroxides in what's known as the Fenton reaction.
The Fenton reaction.
Right. And this reaction throws off these highly toxic hydroxyl radicals. They violently rip electrons away from the lipids that make up the cell membrane.
Wow.
Yeah, it creates this massive destructive chain reaction. If a cell can't stop this free iron, the membrane effectively rusts, it shatters, and the cell just dies.
And for a long time, the scientific community assumed we basically understood how tumors interacted with this process. But the researchers behind this study, they wanted to watch this happen dynamically.
They wanted to see the whole life cycle, yeah.
So they used two different mouse models of liver cancer, hepatocellular carcinoma. And they used this to track the disease from a totally healthy liver all the way to a terminal state.
And using two models is a really crucial detail here. I mean, they didn't want to just rely on some genetic fluke. So first they used a genetic model. It's a specific double knockout called LTSC1 patent.
Okay, and what does that knockout actually do?
It essentially strips away the liver's metabolic brakes. So it mimics how tumors naturally arise from severe metabolic dysfunction.
Got it.
But then they back that up with a chemical model. They expose the mice to carcinogens like DEN and carbon tetrachloride to mimic, real-world environmental liver damage.
Okay, so they tracked the disease progression in both of these distinct models. And this is where they hit this massive, just mind-blowing aha mystery. They observed this distinct biphasic pattern.
Yeah, biphasic, meaning the disease completely changes its behavior in two totally distinct phases.
Right.
In the early stages, when the liver is just starting to get sick and form those precancerous lesions, the markers for that cellular rush are incredibly high. The researchers measured specific molecules called 4-HNE and MDA.
Which, just to clarify for anyone trying to visualize this, 4-HNE and MDA aren't just arbitrary markers. They are the literal broken, oxidized pieces of the shattered lipid membranes.
Yeah, it's the cellular shrapnel.
Exactly. They are the chemical debris of ferroptosis.
Exactly the debris you'd expect to see. And along with those debris markers, the levels of that dangerous free F2 plus iron were just off the charts. The early tumor cells were rusting rapidly. Ferroptosis was happening exactly as textbook chemistry would predict.
But then comes the second phase. Fast forward to the terminal, late-stage cancer, and those exact same debris markers suddenly plummet. The rusting just completely stops.
It just disappears.
The cancer somehow figures out a way to suppress ferroptosis so it can survive, grow, and become lethal.
To survive, the tumor has to hide that massive stockpile of free iron. It has to, pull the fuel source away from the fire.
Wait, hold on. I have to push back here. Iron doesn't just vanish into thin air. It's a fundamental elemental metal.
It sure is.
If the cancer needs to stop this rusting to survive, how exactly is a microscopic tumor pulling off this disappearing act? Where is the iron going?
That is the exact paradox the researchers needed to solve. And to find out how the tumor was hiding the iron, they couldn't just look at the genetics. They had to look at the tumor's real-time chemistry. So they performed untargeted metabolomics.
Okay, before we get to the metabolomics, let's do a quick recap so we don't lose anyone.
Good idea.
So, key takeaway so far. Ferroptosis is this iron-driven cell death, basically cellular rusting. Early liver tumors have really high levels of this ferroptosis, but late-stage tumors somehow shut it down completely to survive.
Right.
So here's a question for you, listening right now. Think about a time you tried to solve a problem by removing the fuel source. How do you think a microscopic tumor actually hides its fuel, this free iron?
Well, to answer that, we go back to the untargeted metabolomics.
Right. And for those trying to picture this, untargeted metabolomics is essentially doing a massive, totally unbiased sweep of the chemical soup inside the cell.
Yeah, you aren't looking for one specific thing.
Exactly. You are taking a complete inventory of every single small molecule, every metabolite present in the healthy liver, and then comparing it line by line to the inventory of the terminal cancer liver.
And sifting through that massive data set, the evidence pointed to one glaring anomaly. The aggressive late-stage tumors had a massive upregulation of polyamines. They were absolutely hoarding one specific polyamine called spermine.
Okay, again, I'm going to jump in. Polyamines. I've seen spermine in biology textbooks, but it's almost always described as a basic structural molecule.
Just boring scaffolding, right?
The scaffolding that hangs out in the nucleus and stabilizes DNA. You're telling me this basic building block is actively hunting down iron?
That was the exact bias the whole scientific community had. We assumed it was just cellular scaffolding. But the metabolomics data was undeniable. The tumors were just flooded with it.
Let's break down how a cell actually gets spermine, because the physical structure of this molecule is the key to everything that follows.
It really is.
Think of the polyamine pathway, like building a train, right? You're adding boxcars one by one. You start with a base molecule called putrescine. That's your single train car.
Okay.
The cell utilizes an enzyme to add a chemical piece to it, creating spermidine. Now you have a two-car train. Then adds one final piece to create spermine. So spermine is the longest train in this specific chemical yard.
And the liver tumors were intentionally building and hoarding that longest train. But this survival strategy comes with a massive, very dangerous catch for the cancer cell.
Oh, there's always a catch.
Always. It involves polyamine catabolism, so the natural breakdown of these molecules.
Because cells are constantly recycling their components.
They are. And when spermine is naturally broken down by the cell, it is chopped up by a specific crew of wrecking enzymes. These are SAT1, SMOX, and PAOX.
Okay.
And when these enzymes dismantle the spermine train, they throw off incredibly toxic exhaust. Specifically, they release hydrogen peroxide and a highly reactive destructive chemical called acrolein,
which is entirely counterproductive for the tumor. Hydrogen peroxide and acrolein are exactly the kinds of reactive chemicals that fuel the Fenton reaction and trigger ferroptosis in the first place.
Exactly. The tumor is essentially trapped in a paradox. It desperately needs the intact spermine to survive the iron, but if that spermine breaks down, the resulting chemical trash triggers the exact cell death it was running from.
So the researchers had to prove this delicate balancing act was actually happening in real time.
And to prove it beyond a shadow of a doubt, they attacked the problem from two different angles. First, they used genetic engineering to create a triple knockout cell line.
A triple knockout.
Right. They literally deleted the genes for those three wrecking enzymes, SAT1, SMOX, and PAOX. So the cell was physically incapable of breaking down the spermine.
And to make sure it wasn't just a quirk of, genetic engineering, they backed it up with pharmacology.
Right.
They used a drug cocktail called the MJP cocktail. That's MDL 72527, JNJ 9350, and pentamidine. This cocktail chemically paralyzed those exact same wrecking enzymes.
And whether they used the genetic knockout or the MJP drug cocktail, the result was exactly the same. Once they stopped the breakdown of spermine, they proved that the accumulated spermine itself acts as an absolute shield against ferroptosis.
Wow.
Yeah, even when they blasted the cells with classic, highly potent ferroptosis-inducing drugs like RSL3 and Erastin, the cells survived.
Which is a huge paradigm shift for oncology. I mean, we've used drugs like RSL3 and Erastin in lab settings for a while, just assuming they directly cause the cellular rust.
Right, that was the standard assumption.
So this paper reveals a massive blind spot. Part of how these classic drugs actually work is by forcing the cancer cell to destroy its own spermine supply. The drugs essentially derail the spermine trains, flood the cell with that toxic exhaust, and force the tumor to burn itself down.
What's fascinating here is it completely changes our understanding of the mechanism of action for these experimental therapies.
Okay, time for another quick recap.
Let's do it.
So, tumors ramp up spermine to survive. But breaking spermine down releases toxic chemicals that trigger cell death. So if you were designing a cancer drug based on this new information, would you want to build up the spermine train, or would you try to derail it completely?
Right. And if you're an oncologist looking at this, you realize that manipulating this pathway is literally a matter of life and death for the tumor. But it still leaves a gaping hole in the story. We know the intact spermine train protects the tumor, but how? What is the actual physical mechanism?
Initially, the logical hypothesis was that spermine was just acting as a standard radical-trapping antioxidant.
Like vitamin E.
Yeah, like vitamin E. The scientific assumption was that spermine acts like a chemical broom, just sweeping up those reactive oxygen species before they can damage the lipid membrane.
But to test that broom hypothesis, the researchers used a highly controlled cell-free system. It's called a Phoenix assay.
A cell-free system, meaning they take all the messy, unpredictable biology out of the equation. It's just pure chemistry in a tube.
Exactly. You put pure lipids in a test tube, you add a chemical oxidant to start the rusting process, and you just watch the lipids break down.
Simple enough.
If spermine is a direct antioxidant, if it is a broom, adding it to that test tube should immediately stop the lipid breakdown.
But I'm looking at the data from the Phoenix assay here, and the spermine completely fails. It does absolutely nothing to stop the direct oxidation of the lipids in that tube.
Nothing at all.
It is a terrible broom.
It failed completely. It is not a radical trapping antioxidant.
Okay, here's where it gets really interesting. If it's not a broom sweeping up the damage, what is it doing? How is it stopping the ferroptosis in the actual living mice?
Well, it isn't cleaning up the fire. It's locking up the arsonist.
Oh, I like that.
Spermine acts as an incredibly potent, highly specific iron chelator. It binds directly to the labile ferrous iron, the F2+N, and the reason it can do this comes down to pure, beautiful, physical chemistry.
Let's go back to our train analogy and look at the actual molecular structure. Putrescine, the single train car, has 2 amino groups. Spermidine, the 2-car train, has 3, but spermine, the longest train, has exactly 4 amino groups.
And in the three-dimensional world of molecular geometry, the spatial arrangement of those 4 amino groups is incredibly critical. They are perfectly positioned to interact with the electron orbitals of a single iron atom.
Right.
Putrescine and spermidine just do not have enough chemical arms to effectively surround and trap the iron.
So if putrescine is like a weak handshake, spermine's 4 amino groups act as this 4-armed molecular bear hug.
A bear hug, exactly.
It locks the free iron away in a chemical cage so tightly that the iron can no longer react with cellular peroxides. The Fenton reaction is physically blocked. The cellular rusting never even gets a chance to start.
And the paper doesn't just theorize this bear hug. I mean, it provides rigorous physical chemistry proofs. They used isothermal titration calorimetry, or ITC.
Which, that measures the minuscule amounts of heat released or absorbed during a chemical reaction, right?
Yes. And when they mixed spermine and iron in the ITC machine, they recorded a highly exothermic reaction. It releases a significant amount of heat.
Meaning what, exactly?
Well, in thermodynamics, that tells us this binding is incredibly energetically favorable. The spermine wants to bind the iron, and it holds on with a very tight grip. And the ITC proved it binds in a perfect 1-to-1 stoichiometry.
1-to-1?
Yeah, one spermine molecule perfectly cages one single iron atom.
They didn't stop at heat signatures, though, which I love. They wanted to actually see the hug happening.
To do that, they used Raman spectroscopy. This technique bounces a laser off the molecule to measure how its specific chemical bonds are vibrating.
Because molecules aren't just static sculptures, they are constantly vibrating and flexing.
Right. So when spermine is just floating around, not holding anything, its specific nitrogen-hydrogen bonds vibrate at a baseline frequency of 1447 inverse centimeters.
1447.
But the exact moment the spermine grabs the iron atom, that vibrational frequency physically shifts to 1453 inverse centimeters.
That is wild. You are literally seeing the mechanical strain on the molecule. You can measure the spermine flexing its chemical muscles to keep that iron locked in the cage.
And they corroborated that with NMR spectroscopy, too. They confirmed rapid spin-spin relaxation, which proves the actual physical reality of those iron-nitrogen bonds.
Wow.
But perhaps the most elegant part of this entire mechanism is its specificity. Spermine is not a blunt instrument. It almost entirely ignores Fa plusage, which is ferric iron, and it mostly ignores copper.
So it's piggy.
Very. It is biologically tailored to specifically hunt down and neutralize Ft plusades, the exact specific flavor of iron that acts as the catalyst for ferroptosis.
It is a perfect custom-fit molecular SACE. If you're picturing this happening inside a tumor cell, the cell isn't trying to clean up the smoke after the fire starts. It is frantically building millions of these microscopic SAFEs to lock away the highly reactive iron before a single spark can catch.
Which brings up the crucial next question for the researchers. If spermine is this miraculous fireproof safe, how does the cancer cell get so much of it? What is the origin story of this superpower?
Let's do our third recap before we get into the origin story.
Good pacing.
So summary. Spermine isn't a broom. It's not an antioxidant. It uses its 4 amino groups to physically trap free Fe2 plus iron in a molecular bear hug, preventing cellular damage.
Right.
So for you listening, imagine you have a highly reactive ingredient in your kitchen. Do you try to clean up the smoke after it burns, or do you lock it in a fireproof safe? That safe is spermine.
Exactly. So back to where the tumor gets all this spermine.
Right, because it's clearly not just relying on the baseline levels a normal, healthy liver cell would use just for DNA stabilization. The tumor is manufacturing this stuff on an industrial scale.
Right. And the researchers traced the biological assembly line all the way back to a master switch. It's an enzyme called ALDH18A1.
ALDH18A1.
Yes. They discovered that aggressive tumors upregulate this specific enzyme to drive an alternative glutamine-dependent metabolic pathway.
Okay.
This allows the cell to build massive amounts of spermine from scratch, completely independent of normal cellular controls.
The tumor essentially flips the ALDH18A1 switch, diverts its glutamine resources, and just starts pumping out SAFEs to hide its iron.
Yep.
So naturally, the therapeutic goal becomes really obvious.
Yeah.
What happens if we smash that switch?
They tested that. They used both genetic knockouts of ALDH18A1 and a novel small molecule inhibitor called YG1702.
And without that master switch, the tumor's industrial assembly line just shuts down.
Completely. By blocking ALDH18A1, they successfully depleted the tumor's spermine supply.
And without the SAFEs.
Without those 4-armed molecular bear hugs, the massive stockpile of free F2 plus iron was suddenly unleashed into the cytoplasm. The Fenton reaction kicked off immediately, ferroptosis was triggered en masse, and the growth of the liver cancer in the mouse models was severely impaired.
They literally starved the tumor of its protective SAFEs, and the cancer just rusted itself to death from the inside out.
If we connect this to the bigger picture, though, this mechanism isn't just a weapon to destroy tumors.
Right. This is where it gets crazy.
We can take this exact same biology, flip the context completely, and use it to save healthy tissue from massive trauma.
It is the ultimate double-edged sword. Let's talk about ischemia reperfusion injury, or IR injury. This is a massive issue in emergency medicine and transplant surgery.
Very common, yes. IR injury occurs when the blood supply is completely cut off from an organ. So think during a severe heart attack, an ischemic stroke, or when an organ is harvested for transplant.
Right.
And then suddenly, the blood flow is restored.
And the paradox of IR injury is that, logically you would think the returning oxygen and nutrients would be entirely good for the dying tissue. But it's actually the reperfusion, the sudden return of blood, that causes the most devastating shock.
Exactly, because the returning oxygen hits damaged mitochondria, triggering a massive, uncontrolled spike in reactive oxygen species.
Right.
And at the exact same time, the damaged cells release huge amounts of stored iron. So you have massive ROS and massive free iron meeting in the same place at the exact same time.
It's a powder keg.
It triggers a catastrophic wave of ferroptosis. The tissue essentially rusts to death the moment the oxygen returns.
So the researchers set up mouse models to mimic this exact clinical scenario. They induced severe ischemia reperfusion injury in the liver, the intestines, and the kidneys. The subsequent tissue destruction was massive, mimicking exactly what happens in a human trauma bay.
But then, in a separate group of mice undergoing the exact same trauma, they administered a remarkably simple intervention.
I love this part.
They gave the mice an injection of pure spermine, just 1 milligram per kilogram, shortly before the blood flow was restored.
They intentionally flooded the dying organs with the molecular safes.
And it worked beautifully. The injected spermine circulated into the ischemic tissues, acted as a highly potent iron chelator, and locked up the free iron before the returning oxygen could trigger the Fenton reaction.
Wow.
The spermine protected the livers, the intestines, and the kidneys. It preserved the delicate tissue architecture. It drastically reduced the biochemical markers of cell death and significantly accelerated the organ's overall recovery.
Just think about the implications of that for a second. It is a perfect demonstration of how biological context dictates absolutely everything.
Everything.
Let's do our final mini recap here.
Yeah.
Blocking ALDH18A1 kills liver cancer by removing spermine. Conversely, injecting spermine saves healthy organs from ischemia reperfusion injury.
Beautifully summarized.
So to you listening, think about this. It's the ultimate double-edged sword. You want to starve a tumor of spermine, but flood a dying kidney with it. How incredible is it that one single molecule dictates life and death in completely different scenarios?
It really highlights a massively underappreciated layer of biology. We are looking at metabolic iron buffering.
Metabolic iron buffering.
Yes. Our cells are using small, internally manufactured metabolites to manage and disarm dangerous elemental metals.
And so what does this all mean for you? This isn't just an abstract process happening in a petri dish somewhere. The ALDH18A1 spermine axis is now a prime, tangible target for the next generation of therapies in both oncology and trauma medicine.
This raises an important question, I think. It forces us to reevaluate our entire biological toolbox.
How so?
Well, if a common everyday molecule like spermine, something we've known about for decades and mostly just dismissed as boring structural scaffolding, if that has been secretly moonlighting as a master regulator of iron and cell death, what else are we missing?
Oh, wow.
What other basic, well-understood metabolites floating around inside your body right now are hiding world-class superpowers?
It really makes you wonder how many other cures are already synthesized and sitting inside our own cells, just waiting for us to actually understand the chemistry.
Exactly.
Well, thank you for joining us on this deep dive. Next time you see a rusty piece of metal, I really hope you think about the invisible, highly choreographed battle happening inside your own tissues and those microscopic molecular bear hugs working tirelessly to keep you from rusting away. Keep questioning the invisible mechanisms operating around you, and we'll see you next time.